Hello and welcome back to my YouTube channel. In our previous videos regarding general pathology, we discussed how a foreign agent or a microbe can cause a set of diseases in our body and will lead to dysfunction in the body system. In this video, we'll discuss about how our own body can become our worst enemy and it can trigger an array of diseases if there is dysfunction. This phenomenon is called as autoimmunity. For the ease of the viewer, I have divided this into a 6-part lecture series. In the first 2 parts, in this video especially, I will be covering the basics of autoimmunity and central and peripheral tolerance. In the next part, we will discuss the general mechanisms of autoimmunity, and in the rest of the parts, I will be discussing certain important autoimmune diseases which are important even for exam going students. And as always, doubts, questions and healthy discussions are always welcome because knowledge shared is wisdom gained. So let's begin. So how exactly do we define autoimmunity? Autoimmunity can be first defined pathologically. So pathologically, its definition can be divided into 3 parts. It is a presence of a immune reaction which is very specific to a particular antigen or a tissue. Secondly, this immune response, it is a primary pathogenic reaction. What do I mean by that? This is primary. It is not secondary to any tissue damage. If it occurs due to any tissue damage, if the response is such that it occurs after a tissue damage, it is not autoimmunity. And thirdly, there is no well-defined cause. There is no well-defined cause as to why this reaction occurs. So, these are the 3 subtext of the definition of autoimmunity. Along the same lines, an autoimmune disease is any disease which is arising due to a lack of self-tolerance. Now, what do I mean by self-tolerance? That we will see in the forthcoming videos. So, what is this self-tolerance and immunological tolerance? It is defined as, immunological tolerance is defined as a phenomenon of unresponsiveness to an antigen which is induced by exposure of the lymphocytes to that particular antigen. So what I mean by this is, whenever our lymphocytes are exposed to a particular antigen, in case these antigens are dangerous or self-reactive, there will be an unresponsiveness by the body to that particular antigen. This is what broadly immunological tolerance is. Then what is self-tolerance? In self-tolerance, there is basically unresponsiveness to an individual's own antigens. Broadly, immunological tolerance can be classified into central tolerance as well as peripheral tolerance. Are you a little confused about this definition and the classifications? We'll explore this in the further slides. So, we'll now dive deeper into the world of central tolerance and peripheral tolerance. Since these are important concepts to be understood even for exam going students, please be with me till the end. In order to understand this, we must first understand how the 2 major WBCs, which is B cells as well as T cells, they develop. So T cells, they develop in an organ which is called as thymus, and B cells, they develop in an organ which is called as bone marrow. The tolerance that they develop here is called as central tolerance. Then what is peripheral tolerance? These two, so thymus and bone marrow, are called as organs of maturation. That is the organs where these B cells and T cells mature. Now once they leave the organ of maturation, outside whatever tolerance they develop, that is called as peripheral tolerance. So peripheral tolerance is the tolerance that is developed outside the organ of maturation, that is outside the bone marrow and the thymus, and central tolerance is what they develop within the bone marrow and the thymus. Now that this is understood, we'll explore further about central tolerance of B and T cells and peripheral tolerance of the same. We'll first discuss about central tolerance in T cells. Now, central tolerance in T cell, like we have discussed before, takes place exclusively in the thymus. Central tolerance by itself as a concept involves two parts to it. One is recognizing the self-antigen, and the second part is killing or rendering it harmless. There are two ways by which central tolerance is achieved in T cell. The first is negative selection or clonal deletion, and the second is development into regulatory T cell. So, how does the first mechanism take place? Now, the concept is very simple. Any sort of a T cell, even the ones which are killed peripherally, must first be expressed. T cells are expressed with the help of APC or the antigen presenting cell. But antigen presenting cell does not work alone. It will need something called as MHC, which is major histocompatibility complex. What is this major histocompatibility complex? That I will explain in a further video, but just understand that it is a sort of a peptide system which helps the T cell to recognize the APC. So, this MHC recognizes the APC for the T cell. Now, what will happen is, after it gets presented by the antigen presenting cell, whichever T cell has increased affinity to become a self antigen, it will get eliminated. So, whichever T cell has increased affinity to become self antigen, this will undergo elimination. This is the basic concept of negative selection or clonal deletion. So, the T cell which can become a self antigen and which can become harmful, they will get deleted. Now, a very important protein that helps achieve this negative selection or clonal deletion is something called as AIRE. So, AIRE is also called as autoimmune regulator. When this AIRE undergoes some sort of a mutation, it would lead to a disorder called as autoimmune polyendocrine syndrome. Now that we have discussed about negative selection or clonal deletion, let us now understand what is development into regulatory T cell. So here this mechanism is exclusively for the CD4 plus T cell lineage. T cell lineage is primarily into two parts. One is CD4 plus T cell lineage and the other is CD8 plus T cell lineage. So whenever there is a CD4 plus T cell lineage which can potentially become a self-reactive T lymphocyte, it gets converted into a CD8 plus regulatory T cell. So once it becomes a regulatory T cell, it does not become harmful anymore. It is rendered harmless, like we had discussed prior. Now, how this mechanism occurs is unknown. It's an unknown mechanism. So just to recap, the first mechanism that we discussed was clonal deletion or negative selection, wherein whichever T cell which is presented with the help of the APC plus the major histocompatibility complex, if it is harmful, it undergoes deletion. What do I mean by harmful? If it becomes self-reactive and that is recognized in the thymus itself, it undergoes negative selection or clonal deletion. The second, where the mechanism is unknown, occurs exclusively in the CD4 plus T cells, which develops into CD8 plus T cell, and that is called as development into regulatory T cells. Central tolerance take place in B cell. In B cell, there is only a mechanism called as receptor editing that takes place. So what is this receptor editing? Say for example, you have a developing B cell in the bone marrow. If it recognizes a harmful self antigen, it will undergo certain gene rearrangements. So what happens because of this gene rearrangements? It will become a completely different developing B cell. This B cell will be something completely different. So this B cell will not be able to recognize this harmful self antigen. This is the basic concept of receptor editing. Now what happens if receptor editing fails to take place? If receptor editing fails to take place, then like we discussed before, deletion of that particular B cell will occur. This B cell will be deleted. Now that we have understood how central tolerance takes place in B and T cell, let us understand how peripheral tolerance takes place in T cell. So, peripheral tolerance is nothing but the mechanisms that occur outside the organ of maturation. Like we discussed before, it is thymus in case of T cell and bone marrow in case of B cell. For peripheral tolerance to take place in T cell, there are three major mechanisms. The first is energy. The second is suppression by any regulatory T cell, and the third is deletion by apoptosis. So, we will discuss about each of these mechanisms one by one. So, the first concept that we will discuss is about energy. So, what is energy? Energy is very simple. Energy means no energy. Okay? Energy is nil. That is energy. Okay? So, what happens here? Whenever there is a potentially self-reactive harmful T lymphocyte, they lose energy. That's the basic concept of energy. Okay? How do they lose energy? Let me explain. So in the previous slide about central tolerance, we saw that any T cell, if it has to be expressed, will require the help of APC plus MHC, right? Now in this concept of energy, they will require something else also apart from APC and MHC. What is that something else? It's called as second signals or co-stimulators. Let me explain with the help of an example. Say you have a T cell which is called as CD28. Now this CD28, if it has to be expressed, it will need what, like I said before, APC plus MHC, right? But apart from that, it will need a set of co-stimulators. In case of CD28, these co-stimulators are B1 plus B2. These are the set of co-stimulator. But what happens is, when this B1 and B2, they are expressed lesser, there is decreased expression of B1 and B2, then that would lead to anergy of CD28. So this is one mechanism. So sometimes what will happen is T cells like the CD28, they will receive either decreased signals from co-stimulators like B1 and B2, or they will receive signals from receptors which serve opposite functions to B1 and B2. So whenever there are receptors which serve opposite function to B1 and B2, then these are called as co-inhibitors. So whenever it receives signals from co-inhibitors, that also leads to anergy. Alright. So in this regard, there are two examples that is very important for students. So the first co-inhibitor is B7. B7 is a co-inhibitor for CTLA4, and PDL1 and PDL2 are co-inhibitors for PD1. So whenever there are mutations in this, it can lead to various autoimmune diseases. The next is suppression by regulatory T cell. Now, like we saw in the previous slides, these regulatory T cell, it usually prevents immune reaction against self antigens. What do these regulatory T cell do? They prevent an immune reaction against the self antigen. In this regard, these expressed by the CD4 plus T cells. That is what I am trying to say is, in this regard, the CD4 plus T cells, they express certain T cells, okay, like CD25, the alpha chain of IL-2 receptor, and FOXP3. Now whenever there is a mutation of CD25, it can cause diabetes mellitus or multiple sclerosis. Similarly, mutation in the alpha chain of IL-2 receptor can cause multi-organ autoimmune disease. And mutation of FOXP3 will cause a disease called as IPEX. What is the full form of IPEX? It is a autoimmune disease wherein there is polyendocrinopathy, etc. The third mechanism is deletion by apoptosis. This is a concept that we have already discussed in the previous video on apoptosis. So here what happens is, this T cell will basically undergo deletion. How will it undergo deletion? Via two mechanisms. One is the mitochondrial pathway of apoptosis, and the next is the death receptor pathway of apoptosis. Now these are concepts that we have already discussed before. So but just to recap, what happens in the mitochondrial pathway? There is unopposed BIM production, which is pro-apoptotic, without the anti-apoptotic proteins. That is, no anti-apoptotic proteins are there. Which are the anti-apoptotic proteins? They are BCL2 and BCLX. In the death receptor pathway, what gets involved? The FAS and FAS ligand system. So what happens in this FAS ligand system? they will tend to trigger the CD95. Further details as to how apoptosis occurs can be seen in my video which I have discussed prior. So how does peripheral tolerance take place in B cell? In B cell, peripheral tolerance takes place through the mechanism of energy, wherein, like we just discussed, because of certain mechanism, the B cell will fail to respond. So I hope you enjoyed this video. In this video, we discussed about the first part of autoimmunity, wherein we defined autoimmunity, and we also discussed about immunological tolerance and its two major components, central and peripheral tolerance. The failure of central and peripheral tolerance will lead to autoimmune diseases. So that will now be followed by part 2 video, wherein we will discuss certain general mechanism because of which autoimmunity occurs. If you like this video, please like, share and subscribe to my YouTube channel, and as always, comments, healthy discussions and questions are always welcome in the comment section below. See you in the next video.